Good morning, class. Today I'm discussing with you first year BSc Biotechnology second semester, paper 2, microbiological methods, 8 marks important questions. Okay, so the first question is here: what is microdilution? Explain its methodology. Broth microdilution is a method used to test the susceptibility of microorganisms to antibiotics. When you take this microdilution methods, this can be divided into two ways, that is broth dilution, other one is the agar dilution. Okay, so we can go through this broth dilution and you can write about in detail broth dilution procedure and all those things. Okay, you can take screenshot and go through. I'm not explaining in detail as we already gone through in the class. Okay, so this is about micro dilution and further preparation of antibiotic stock solution, then preparation of antibiotic dilution range you need to write, and further please also go through this diagrammatic representation of broth dilution method for measuring minimum inhibitory concentration of antibiotics. Okay, in this first you are going to obtaining isolated colonies of bacterial strain to test. Okay, this is the first one. Then second, combine 4-5 colonies and culture overnight in rich media broth. Then you'll go for the plate oligcoat. Then after overnight incubation, you are going to check cultures for growth. Okay, so here you are going to understand about the broth dilution method information you must write. Okay, this is about the preparation of agar dilution plates. Further, can also talk about preparation of inoculum, then incubation, then reading and interpreting results. Okay. Also, if time permits, you can write about advantages. Then spread plate method, as you all know, the spread plate method is also a method for the isolation of the microorganisms or counting of the individual microorganisms, where you can see the distribution of colonies very evenly. Here, the spread plate technique is used for enumeration of enrichment, screening, and selection of microorganisms. In this, the culture is uniformly spread over the surface of an agar plate, resulting in the formation of isolated colonies distributed evenly across the agar surface if the appropriate concentration of cells is plated. So, in this spread plate method, as we are spreading the inoculum over the culture media, which gives the separated colonies, that will be easy for the counting. Okay, please go through all this information and write the relevant information. Okay. Further, you should also write about the spread plate method, diagrammatic representation flow chart. Okay, so here you could see the bacterial dilution poured onto the solid medium, then spread sample evenly over the surface by using L-shaped glass rod or triangle-shaped glass rod. Okay, then plate incubated until bacterial colonies grow on the surface on the medium. As you could see here, individual colonies can be counted and we can observe this particular through microscope. Individual colonies can be counted. Very separated colonies can be seen over through this spread plate method. And also you can focus on here material required to perform the spread plate technique, and here you could see the procedure, then advantages over other methods and limitations of spread plate method. Okay, next, sorbitol assay. In this assay, the sorbitol is oxidized to fructose with the proportional development of intense color with an absorbance maximum at 560 nanometer. Here, the assay is useful for the range of 0.1 to 10 and mole of sorbitol per sample. Here, interestingly, sorbitol can be used as screen for the 0154H7 strain of E. coli, since this strain is one of the few strains which cannot metabolize sorbitol. Okay, further in detail, you please go through about the sorbitol information. Okay, even this is important for your examination point of view. You can kindly focus on sorbitol assay. Whereas in this method, the media will be provided with sorbitol as osmoprotectant for the fungi, whereas in the control, there is no sorbitol. Okay, this will detect the MIC shift to higher level as the sorbitol is protecting the fungal cells from the antifungal compounds. And it is a negative test done to check the antifungal activity of the compounds. Okay. For further more information, you can also refer your notes, whatever is given. And here this is about the applications of sorbitol assay, and this is more information about sorbitol. Okay, if possible, you write this one. Otherwise, previously whatever showed, no, that you can write. You can take screenshot and go through that one. Next question: define Beerus and Lambert's law. Write short note on spectrophotometer. Okay, write about Beerus law and Lambert's law. Okay, and write their formula together. Okay, so that will define about Beerus law and Lambert's law. Okay, please take screenshot and refer this information. Okay, here I've mentioned about the spectrophotometer. Okay, because you need to explain about spectrophotometer, which is a laboratory instrument that is used to measure electromagnetic energy at specific wavelengths of light. Okay, it can measure the concentration of known substances in a solution by passing a light through the substance measuring the light intensity as a function of wavelength. Okay, please refer all this information and write about spectrophotometer. As there is specifically not mentioned about the spectrophotometer work or application, but have mentioned in detail about the spectrophotometer work and further also about the applications. Okay, please pause this video and go through this spectrophotometer work information. And here I mentioned here spectrophotometer applications. Okay, so this is all about the applications of spectrophotometer. Okay, take screenshot or go to this one, pause this video and please have a look. So this is all about the spectrophotometer. Next, describe disk diffusion technique. And this method used to determine microbial susceptibility to antibiotics, in which filter paper disc containing known concentration of antibiotics are placed on the agar surface that has been previously inoculated with the bacteria of interest. Okay, in diagnostic laboratories, with all this diffusion test will be used to determine the susceptibility of clinical isolates of bacteria to different antibiotics. As you can see, this is the before growth. This is the culture plate. To this culture plate, we have already inoculated the microorganism, okay, in a swabbing method. Okay, on top of the bacterial spread, we have placed the different antibiotic disc. Okay, this is the agar media and spread on petri dish with the inoculum. Okay, grow after the growth, we'll be able to observe this like this kind of view. And this is the no bacterial growth. So this tells us about the zone of inhibition. Okay, where this is the zone where this antibiotics has inhibited or killed the growth of this bacteria. And this is here you can see this is the bacterial growth, and this antibiotics is unable to kill this particular bacteria, so there is no any kind of zone of inhibition here. Little zone of inhibition, and here you can see the more amount of inhibition. So please talk here about disk diffusion test and their principle. Go through all these things and you need to write about the disk diffusion method. Okay, in detail I have mentioned here, or take screenshot or pause the video and go through this disk diffusion method. If you have received the notes, you can refer your notes also. Next, last question, briefly explain antimicrobial activity. Okay, antimicrobial sensitivity test of the antimicrobial activity. Here you can also take the example of this diffusion method and you explain this antimicrobial sensitivity test and maybe antibiotic sensitivity test. Okay, the same further information. So this is the pictorial representation of the agar plate with antibiotic disc showing zone of inhibition. This also you can take for the antimicrobial sensitivity test and even disc diffusion technique. Okay, here you can see the zone of inhibition and bacterial growth here. Based on the comparisons of zone of inhibition ZOI on the plate, the standard values of the bacteria tested were determined to be resistant or intermediate and susceptible to different antibiotics present. So this is the zone of inhibition, and this is the antibiotic, and this is the bacterial growth or maybe a bacterial lawn, and this is the intermediate between these two zone of inhibition, and this is the resistance. Okay, this antibiotic is not able to kill the bacteria, so this bacteria is resistant towards the antibiotic, and this is the susceptible. Okay, so this is about the antimicrobial sensitivity test or antibiotic sensitivity test. So all these questions you need to focus. Thank you everyone for watching this video. I, Dr. Pratibha K, signing out for the day. Stay tuned with me. Thank you.